So, yes, good evening, everyone. This side Dr. Abhijeet, and today, we have initiated a 90 days foundational life class for university exams where we are going to cover nucleic acids and nucleotides biochemistry part 1 today. So, I will be discussing all the important questions, all the potent questions that I feel are important for the upcoming prof exams that will take place in November only, right? So I will be covering all the previous 10-year questions, the previous year topics, right? In the form of question and answer format, I will be discussing the entire topic and I will be telling you how to write the answer in the exam. Okay, to gain like maximum marks, right? So, yes. So here we are rapidly revising all the questions, okay, in a very crisp, concise, and conceptual manner, right? And it is like beneficial for even the students appearing for the upcoming NEET PG exam and the FMG exam as well. Guys, today I will be discussing the session within like three parts, the long essays, short essays, and the short answer format. The long essay is basically the 10 marks question, right? In which we have to cover each and every particular detail and how to write this, I will be telling you. So I will be discussing the DNA and RNA today, right? The components of DNA, RNA, right? Okay, and different types of RNA also. Short essays, basically, they are either 8 marks, 1 or 2 questions, or 5 marks majorly, right? So I will be discussing the purine and pyrimidine bases, right? Even I will be discussing the Watson and Crick model of DNA, right? And in the short answers, I will be discussing the nucleoside, the nucleotide with examples. I will be discussing the types of RNA, and I will be discussing the complementary base pairing rule. Okay, so let us just without wasting time, let us just start. But before that, guys, why to choose Oracle? You already know that we have the recorded sessions. Once you are not attending the classes live, you can automatically go on to the particular channel on YouTube, or you can just subscribe to the app and have the look at the recorded classes later on whenever you have time. Live classes, we are daily conducting live classes from 6 to 10 p.m. on a daily basis, and you can just have an access to all the live classes that are being conducted. Even guys, the practical sessions, no, every student wants to know, review the practical series before going to the exams. Multiple times if you do also, still you need an extra edge. So here comes the role of the Oracle, guys. We have already like the recorded practical sessions before. Okay, apart from that, guys, 1 month before, if you ask me, shall I go for the textbook? So I will say no. Why? Because we have less time in that. You have to cover all the important major topics. And we have this express exam module, which covers the entire topic under 20 hours. So that is something you can just go on for it. And the most important and the most core feature of the Oracle app is this hard copy book set. Guys, you can order these books and this will be delivered to your home, right? And these notes are basically prepared keeping in view the real question and answer format. Okay, and yes, all the important previous year questions, the past 10-year questions are kept in this, so you can just go on for it. Now we have the recorded classes. We have already told you, you can just anytime watch these recorded sessions which are available on the YouTube. Apart from that, the live classes we are conducting daily from 6 to 10 p.m., right? The express exam module, which covers everything under 20 hours. And one-to-one doubt clarification. Again, this is very, very important, guys. You can always book a session wherever you have doubts regarding the topic or regarding the subject with that faculty, and you can just get all your doubts cleared in one go, right? Multiple times you can ask your doubts, right? Even during the live sessions, if you are not, just go for a session book. The management will always help you in this, right? The hard copy book set we have already told, and the Q bank, guys, we have already discussed from basic to advanced level, right? The past 10-year questions, okay, whether it is NEET PG, INI-CET, FMG, okay, and the past 10-year questions in the biochemistry exam that is conducted all over India, no, in various universities. So we have like 2020 MCQs that are being asked that are also like covered. Okay. So yes, we have the practical sessions, guys. The one which I've already told you, they are being recorded. You can always go on for these practical sessions and like, yeah, it will help you to rememorize everything. Now, the basic subscription plans, guys, we have a foundation plan for 3 months, right? We have advanced learning plan, we have complete learning plan, and we have ultimate MBBS plan for 4 years. For additional discounts, guys, you can use my code Dr. Abhijeet 20, right? Now, guys, let us just try to discuss the various structure type. Okay. Now we are going to start with a 10 marks question. We are going to start with the 10 marks question. Just a minute. Huh. So the very first question, which is of 10 marks, it says start, explain the structure, types, and functions of DNA and RNA. Covered the structural components of DNA and RNA, and you already know the types of the RNA, mRNA, tRNA, rRNA, and their respective roles in genetic information, storage, protein synthesis, regulation. So these are the points that we have to actually cover. And yes, now guys, you already have, okay, first let us just talk about the DNA. Now, you guys are aware that DNA, it is also called as deoxy. Okay. DNA, it is also called as deoxyribonucleic acid. Deoxyribonucleic acid. And you guys already know that it is a double-stranded molecule. Okay, it is a double-stranded molecule. Now, this is something which is made up of nucleotides. It is made up of nucleotides. Nucleotides. Now, guys, you already know the nucleotides. If we just talk about the structure, guys, the nucleotides, it is basically made up of three components, right? The nucleotides, they are the building blocks of nucleic acids. Now, when I say nucleic acids, guys, I'm talking about DNA and RNA. So I'm talking about DNA and RNA. These are nothing but the nucleic acids, and nucleotides are nothing but building blocks of nucleic acids. And these nucleic acids, guys, you already know either they are DNA or RNA, deoxyribonucleic acid or ribose nucleic acid. Now, nucleotide, it is basically made up of three components. Three components. We have the nitrogenous base, the nitrogenous base, the pentose sugar, and the phosphate group. The phosphate group. So we have nitrogenous base, the pentose sugar, and the phosphate group. Now, now, guys, whenever we talk about the nitrogenous base, you guys are very much clear this, know from +2 that we have like four nitrogenous bases over here. We have adenine, we have thymine, we have cytosine, and guanine. Adenine, thymine, cytosine, and guanine. right? So these are the 4 nitrogenous bases that are like present in the DNA, right? So yes, now we have the pentose sugar. And guys, over here, remember that adenine and guanine, okay, nitrogenous bases are nothing but purines or pyrimidines. Purines and pyrimidines. Now, again, every time in my lectures, I always say how to learn this. All girls are pure. All girls are pure. This is what I always say, all girls, right? So you guys have to remember it is adenine along with guanine, right? Adenine and guanine, right? Now, adenine is nothing but 6 amino purine, and guanine is nothing but 2 amino 6 oxopurine. 2 amino 6 oxopurine, right? 2 amino 6 oxopurine. Other guys, the pyrimidines. Pyrimidines are nothing but basically you can remember by the help of mnemonic cut, right? So cut, it is cytosine. We have uracil and we have thymine. So guys, remember that the thymine, it is seen in DNA only. Very, very important. It is seen in DNA only. And uracil, it is seen in RNA only. It is seen in RNA only. Okay, it is seen in RNA only. So that is something you have to remember. Now, coming on to the pentose. Now, coming on to the pentose sugar. Now, guys, remember that the pentose sugar, okay, if we talk about the case of RNA, okay, it is ribose sugar. It is ribose sugar. Or if it is, yeah, or it can be deoxyribose sugar. Now, what is the difference, guys? The difference is only in the orientation of OH, right? It is only in the orientation of OH, right? So ribose is basically present in RNA, and deoxyribose, it is present in DNA. Okay, it is present in DNA. Now, guys, remember we have the phosphate group. We have the phosphate group, guys, and you can remember that the nucleotides can have one phosphate. Okay, they can have one phosphate. Okay, they can have two phosphate. They can have three phosphate. Okay, so remember that for one we use the term mono, two it is di, and for three it is tri. So, or anyways, we have adenine, adenine combined with sugar. Okay, adenosine. So we have AMP, adenosine monophosphate. Adenosine monophosphate. Okay, we have ADP, adenosine diphosphate. We have, okay, we have adenosine monophosphate, adenosine diphosphate, adenosine triphosphate, right? So that is something, guys, you have to remember over here. So this is the basic structure that you have to remember. Okay, now. Hmm. Now, again, in this case, guys, you also have to remember some more important parts, right? So remember, DNA, if I just want to like describe in nutshell, so it is a polymer of deoxyribonucleotides. It is a polymer of deoxyribonucleotides. And what are the nitrogenous bases present? We have already discussed this. The nitrogenous bases which are present over here, it is, yes, adenine, guanine, cytosine, and thymine. Again, I've told you uracil, it is present in RNA. Adenine, guanine, cytosine, and thymine. Okay. So basically the deoxyribonucleotides, they are held together. Okay. They are held together by a linkage, guys, that is called as 3' to 5' phosphodiesterase linkage. Phosphodiesterase linkage. Phosphodiesterase linkage. They are held together by 3' to 5' phosphodiesterase linkage. Right? So there are many forms of DNA, guys. We have B form, we have Z form, we have A form, right? So that is something you have to remember. Now, again, if we talk about more about the structure of the DNA, if we talk about more about the structure, okay, we have to discuss the types also of DNA. Okay. So if we talk about the types of DNA, we have like very like there are many different forms of DNA, guys. And apart from that, if we talk about the major ones, we have B form, okay, we have A form, and we have Z form of DNA. Z form of DNA, right? So now what is the difference, guys? The B form, it is right-handed double helix. And remember, guys, it is having 10 base pairs. How many base pairs per turn? 10 base pairs per turn. Very important. It is having 10 base pair per turn. Now, even the A form, it is also right-handed double helix, and it has 11 base pairs per turn. The Z form is the one which is left-handed, and remember it is having 12 base pairs per turn. These are all important MCQs as well, guys. You can just remember the B form, the A form, and Z form, and you have to remember the base pairs per turn, 10, 11, and 12 respectively, right? So now if we talk about the structure of the DNA, okay, we have discussed the composition, how the DNA is made up of. Now, coming on to, yes, the basic structure, guys. Remember that DNA, it is a right-handed double helix, right-handed double helix. And remember, guys, it is consisting of two twisted, yes, polydeoxyribonucleotide strands. Okay. So we have two strands, guys. The double helical structure is there, right? One running in from 3' to 5' direction, right? And one running from 3' to 5' direction. Again, the rule says that they are anti-parallel in nature, the one which you already know from +2, right? And right, it is right-handed double helix structures. Remember that they are parallel to each other. These two strands, guys, they are parallel to each other. They are anti-parallel. So yes, they are anti-parallel. Okay. So actually, it's just like the two roads, guys, which are having opposite traffic directions, right? So double helix, remember, guys, it is having a width of 20 Armstrong, 10 raised to power minus 10. Okay. And a turn, okay. Turn is also called as a pitch, guys. It is having like pitch of 34 Armstrong. 34 Armstrong. Okay. And remember, guys, the base pairs. Okay, we have already discussed we have 10 base pairs, right? So we have, yes, with 10 base pairs, right, of nucleotides. Of nucleotides, right? Now, guys, you already know that Chargaff's rule is applied over here, right? A equals to T and G equals to C, right? So we have the bond pairing rule that a adenine binds combines with thymine with two double bonds, and guanine combines with cytosine with three double bonds, right? So remember, guys, what is the function over here that you have to remember? The function that you have to remember that the DNA basically stores the genetic information. Okay, it stores the genetic information, and remember, it serves as template for replication. We know the central dogma of molecular biology. The central dogma of molecular biology states that from DNA, two DNA is made, right? From DNA, DNA is made, then it is converted into RNA, then it is converted into proteins. This is the central dogma of the molecular biology. From DNA, RNA is made. This is called transcription. And from RNA to proteins, it is called translation. And from DNA to DNA, it is called as, yes, the replication, right? So it is serving as a template for replication also. Template for replication. Okay. And it is responsible for the inheritance of traits. Okay, whatever traits they are passed from one generation to another, right, from the parents to the offspring, so they are basically responsible for the inheritance of traits also, right? So this is something that you have to remember over here, right? Now, hmm. So I can basically say that it is the basis for heredity, or I can simply say the chemical basis for heredity. Chemical basis of heredity, guys. Chemical basis of heredity, right? So that is something uh, you have to remember. The central dogma, the acting as a template for replication, it contains the genetic information, the inheritance of traits. So that is all regarding the functions of the DNA. Now, coming on to the, yes, the RNA. Coming on to the RNA. RNA, remember, it is a polymer of ribonucleotides. Polymer of ribonucleotides. Now, over here, you guys already know, if we talk about the pentose sugar, you already know it is ribose. Pentose sugar, it is ribose. Bases, the nitrogenous bases, guys. Nitrogenous bases, I have already told you, we have adenine, guanine, cytosine, and uracil. Thymine is not present in the DNA. So we have adenine, guanine, cytosine, and uracil. Okay, RNA generally, guys, the RNA, it is single stranded. If we talk about the structure, guys, it is generally single stranded, right? It is single stranded. Now, if we talk about the types of the RNA, remember there are three major types of RNA. We have three major types of RNA. Remember, we have the transfer RNA. It is also called as the tRNA. Then we have messenger RNA. Messenger RNA. It is also called as the mRNA. We also have the ribosomal RNA. That is called as the rRNA, right? Now, these are the major forms of the RNA. But if we talk about the minor types of RNA also, the minor types of RNA, we have, yes, heterogeneous nuclear RNA, heterogeneous nuclear RNA. Okay, that is called as hnRNA. we have small nuclear RNA. Small nuclear RNA that is called as snRNA, right? So heterogeneous nuclear RNA, guys, remember that they are the ones which are precursors of mRNA. Okay, they are precursors of mRNA. The small nuclear RNA, basically, they help in conversion of heterogeneous nuclear RNA, that is hnRNA, to mRNA. To mRNA. Very, very important. So these small nuclear RNA helps in the conversion of hnRNA to mRNA. mRNA precursor is hnRNA. From hnRNA only and then only mRNA will be formed. And the one which is helping in the conversion of hnRNA to mRNA, that is the small nuclear RNA. That is the small nuclear RNA. Now, coming on to the transfer RNA, guys, we have to discuss each type in detail. Yes, coming on to the transfer RNA, that is the tRNA. Transfer RNA, right? So remember, guys, that transfer RNA, it basically consists of 71 to 80 nucleotides. Very, very important. It consists of 71 to 80 nucleotides. Okay. Molecular weight, it is approximately, if we talk about the molecular weight, it is approximately 25,000 Daltons, right? So remember that there are 20 types of tRNA. Very, very important. There are 20 types of tRNA corresponding to 20 amino acids. 20 amino acids, okay, which are found in proteins, right? So remember the structure of the transfer RNA. It is basically in the form of a clover leaf-like structure. Okay, the clover leaf-like structure. The clover leaf-like structure, the picture which you have already seen in your class. Yes, this kind of a picture. Let me just try to draw it more nicely. Right, the clover leaf-like structure. So it is having various loops, right? So we have this clover leaf-like structure. Okay, so the structure is this like this, right? So we have the T loop over here. We have the D loop over here, right? We have the amino acid attachment site. Amino acid attachment site. Okay. We have the anti-codon loop. Anti-codon loop. We have the anti-codons over here. Anti-codons over here. And let us suppose this is the mRNA. This is the mRNA. And we have the codon over here. We have the codon over here. Codon, for example, we have, okay, let us say GAG. And what will be the anti-codon? Yes, yes. Okay. What will be the anti-codon? It will be C. You already know guanine binds with cytosine and adenine binds with uracil. So we have U over here, C over here, and C over here, right? So remember, guys, we have, okay, we have, if we talk about the structure, we have like arms. We have different arms. We have acceptor arm, or it is also called as CCA arm, right? So basically it is having a CCA sequence from 5' to 3', okay, where the amino acid binds, right? We have anticodon arm, okay. So anticodon is the arm which like holds, you already know the three anticodons, which are no help in recognizing the codons from the mRNA, right? Codon and anticodon, guys, always you know they are complementary to each other. We have D arm, the D loop, guys, you can see it is normally, guys, due to no dihydrouridine presence. Then we have T, okay. So basically it consists of okay, T epsilon T C arm, right? It contains a sequence of T, guys, pseudo uridine, that is denoted by this. It is called as pseudo uridine, and we have C, that is the cytosine. And we have the variable arm also, right? So yes, base pairing rules, guys, you already know. And what is the function of this transfer RNA, guys? It carries amino acid to the ribosomes. It carries amino acid to the ribosomes. Okay, it carries the amino acid to the ribosomes during protein synthesis. During protein synthesis. Okay, so yes, coming on to the rRNA. rRNA. Now, rRNA, it is also called as the ribosomal RNA. It is also called as the ribosomal RNA, right? rRNA, it is also called as ribosomal RNA. Now, it contributes nearly about 60 to 70% of the total RNAs of the cell. Total RNAs of cell, right? So most of the rRNAs, basically, they combine with protein and together they exist as ribosomes. Together they exist as the ribosomes. Ribosomes are nothing but they are the nucleoprotein structure. They are nothing but the nucleoprotein particle. And remember, guys, the nucleoprotein particles, okay, the ribosomes have two subunits. Two subunits, a large subunit and a small subunit. A large subunit and a small subunit. Okay, if we talk about the prokaryotic ribosomes and the eukaryotic ribosomes. The prokaryotic ribosomes are 70S ribosomes. Okay. So they are made up of 50S plus 30S. Don't go with the maths. It is 50S plus 30S. Okay. And eukaryotics are 80S ribosomes. They are made up of, yes, 60 and 40. 60S and 40S subunits. The larger being the 60, the smaller being the 40S subunits. Right? So that is you have to remember. So association of these subunits basically they help during protein synthesis. So the function, guys, you have to remember they are factory of protein synthesis. They are factory of protein synthesis. Okay. So basically on ribosomes, mRNA and tRNA interacts to translate the codons present in the mRNA. Okay. So yes, whenever we'll discuss the transcription wala part, we'll be like getting more about amount of like info on this. Coming on to the other type of RNA, guys, that is the mRNA. That is the messenger RNA, guys. The messenger RNA, it is okay. This mRNA, it is synthesized from the template strand of DNA. From the template strand of DNA. Now, guys, you already know the central dogma of molecular biology. From DNA, what is made? DNA only replication. Now, from DNA, the RNA which is formed, it is called as messenger RNA. So this RNA, it is formed from DNA. So it is using the strand of the DNA, okay, that is called as the template strand, so that the RNA can be synthesized, right? So this is called as transcription, right? So RNA is formed keeping in view like the codons from the DNA, right? So mRNA is basically synthesized from that, right? Now, mRNA, if this is a template strand, remember the mRNA basically, which is synthesized, it is complementary. It is complementary to the template strand of DNA. Template strand of DNA. Okay. So you have to remember. So the genetic information which is there, we have already discussed that the DNA, it is basically a carrier of the genetic information, and whatever genetic information is there in the DNA, it is transferred into mRNA, right? It is transferred into or transcribed into mRNA. So remember the genetic message encoded in DNA is transcribed to mRNA. Is transcribed to mRNA, right? So yes, mRNA basically carries the message from the DNA. Okay. And remember, the DNA, guys, you already know it is present in the nucleus, and basically it is carrying the message present in the nucleus to the ribosomes present in the cytoplasm for protein synthesis, right? So that is something you have to remember. Okay. So yes, the major parts will be like more discussing when we'll be discussing the transcription wala part. So that was regarding in short about all the important points that we have to just remember in case of DNA and RNA, their basis, their functions, right? The respective roles in genetic information, right? So that is like you all have to take away the important points over here. Now, question says, describe the structure and significance of purine and pyrimidine bases. Include a description of the molecular structure and how these bases form the foundation of the nucleotides. Now, guys, this is yes. So let us just try to discuss the basis wala part over here. Hmm. Now, firstly, guys, you should know purines and pyrimidines. Purines and pyrimidines. Now, purines are the ones which we already know that all girls are pure. Okay. All girls are pure. Now, from A, you already know we have adenine. And from G, it is guanine. And pyrimidines, remember by the help of mnemonic CUT, we have cytosine, uracil, and thymine. Right? So these are the purines and these are the pyrimidines. One exception that I have already told you guys that uracil it is found in RNA only and thymine it is found in DNA only. Now guys, just try to see over here. Now, you guys already know how to draw a purine, guys. It's very, very, very simple, guys. Just try to draw a benzene ring, right? See, bonding is not necessary in biochemistry. Keeping the valency is not necessary in no over here. Now, guys, what you have to do? Hmm. You have to start the numbering from here. 1, 2, 3, 4, 5, and 6. And obviously, you have to draw, okay, one more structure, just like this. Let me just try to draw. Just draw a benzene ring like this and just try to draw the other structures as well. So yes, I'm not drawing the nitrogen here right now. Let me teach you how easy it is actually. Hmm. Now, guys, just try to see all the odd positions, right? All the odd positions, they are being okay. So this is 6, then this is 7, this is 8, and this is 9. All the odd positions basically they occupy nitrogen, okay? Except the okay, the fifth wala part, okay? So first will have nitrogen, the third will have nitrogen, then yes, the fifth won't have nitrogen, guys, right? All odd positions, all odd positions basically they have nitrogen, okay, except fifth. Seventh is an odd number, it will have nitrogen, and ninth is an odd number, it will have nitrogen, right? And guys, if we are if we are drawing the structure of adenine over here, if we are drawing the structure of adenine, so we already know adenine is nothing but 6-aminopurine. 6-aminopurine. So at sixth position, we will have one NH2 group. We will have one NH2 group. right? So this adenine is nothing but 6-amino purine. 6-amino purine. Guanine is nothing but 2-amino 6-oxopurine. Again, it is very, very easy. Again, try to draw the same structure, guys. Okay, try to draw the purine, right? Try to draw the purine. So all odd position 1, 2, 3, 4, 5, 6, 7, 8, and 9. All odd positions will be occupied by the nitrogen, except the fifth, right? And it is 2-amino, guys. It is 2-amino. Now you already know at second place there will be an amino group, right? NH2 group. 6-oxo. At sixth position there will be an oxygen attached. So it becomes guanine. Okay, it becomes guanine. 2-amino 6-oxo purine. 2-amino 6-oxo purine. Right? So these are the structures regarding with the purines. These are the structures regarding with like relation with the purines. Very easy to draw, guys. You can just remember by the help of the names. Now coming on to the pyrimidines, guys. Pyrimidine is a single ring, guys. Okay, pyrimidine is a single ring. So yes, if we just try to draw the rings, guys. Okay, if we try to draw the cytosine, I'll draw one and rest you can just practice from the home. So it's just a single benzene ring, guys. Okay, it's just a single benzene ring, guys, right? And the numbering over here will be started from here 1, 2, 3, 4, 5, 6, right? So, yes. So, all the odd positions, the nitrogen will be over here, right? The nitrogen will be at the third place, right? So, all the odd positions, one and third, except fifth. All odd positions except fifth will have nitrogen, right? So we will also have this, yes, the NH2 group over here in the cytosine at fourth position. Okay, so 4-amino pyrimidine, right? So similarly you can just draw for thymine and uracil as well. Now coming on to the we have just drawn the structures over here. Okay, so amino group, it is basically present at the fourth position, right? In cytosine. Okay. Now, again, guys, if you just want to remember thymine also. Let me just try to draw thymine. Thymine, it is similar to cytosine, except okay, it has a methyl group. It has a methyl group at, remember, fifth position. Fifth position. It has a methyl group at fifth position, right? It has a methyl group at fifth position, right? And yes, cytosine is also having this carbonyl group, this oxygen. Yes, over here as well. Okay. So remember, it is just similar to like this structure only. Let me just try to draw you for you people. So 1, 2, 3, 4, 5, and 6. All odd positions will occupy nitrogen, except the fifth one. The second will have the carbonyl group. The fourth will again have, it is similar to cytosine. So it is having one NH2 group. Okay. So it basically is similar to the cytosine, right? Except the methyl group, it is present at fifth position. The methyl group, it is present at fifth position, right? Uracil basically similar to thymine but lacks methyl group at fifth position. Okay, uracil lacks CH3 group at fifth position. At fifth position, right? Now, now, what are the functions, guys? What are the functions? So remember, guys, the basic function, it is, yes, the ATP, right? The ATP generation. ATP, you already know it is the energy currency of the cell. It is the energy currency of the cell. Okay, it is the universal carrier of the energy within the body. Okay, ATP is required for muscle contraction and everything, right? So this adenosine, guys, obviously adenosine will be having this adenine component, otherwise adenosine will not be formed, right? Adenosine triphosphate, ATP, it is having that particular base, the nitrogenous base in the form of purine, right? So apart from that, they are all having something called as the genetic, they store the genetic information. Okay, they store the genetic information, right? The purine and the pyrimidine bases, basically they encode the genetic information. Okay, you already know the base pairing rule, also complementary pairing. Okay. Yes, adenine with thymine and guanine with cytosine, or adenine with thymine or uracil, right? Apart from that, they also have the regulatory functions, guys. They also have the regulatory functions, and you already know they act as single signaling molecules. We already know cyclic AMP, adenosine monophosphate, right? They help in enzyme regulation, they help in metabolic pathways that we'll be discussing in the hormonal section as well, the pathway of hormones and everything, right? So that was regarding the this particular question. That is, yes, the structure and the structure of these purines and pyrimidines, and like what are the functions associated with it. Coming on to the next section, Watson-Crick model of the DNA. Watson-Crick model of the DNA. Now, we have already discussed this, guys. It is a double helix structure. All the points I'm just writing in short because we have already covered it before. It is a double helix structure. Double helix structure. Remember that it is having like a twisted ladder kind of a structure, right? A twisted ladder kind of a structure. They run in opposite direction, 5 prime to 3 prime and 5 prime to 3 prime like this, right? Anti-parallel, anti-parallel with one orienting from 3 prime to 5 prime and the other one orienting from 5 prime to 3 prime, right? So basically, this helix consists of no, the sugar, the pentose sugar, and you already know it is the deoxyribose sugar. Okay, it also contains phosphate groups. Okay, this phosphate forms the backbone of the DNA, right? The sugar and the phosphate, they are linked together by the phosphodiester bonds. Phosphodiester bonds. Okay, we have the nitrogenous base, we have the sugar, okay, and we have the, yes, the phosphate. So sugar and phosphate, they are linked together by the phosphodiester bonds, right? Even the base pairing rule, they have also already told the base pairing rule. So basically we have adenine, yes, basically pairs with thymine with 2 hydrogen bonds, and guanine pairs with cytosine with 3 hydrogen bonds, right? So that is something you have to remember. The double helix has width of 20 Armstrong, that we already know. Okay, pitch, you already know it is of 34 Armstrong, and with containing of 10 pairs of nucleotides. They contain 10 pairs of nucleotides, right? So that is something you have to remember in short about all the Watson Crick part, right? Now, guys, you already know what is the difference between a nucleoside and a nucleotide. Nucleoside and a nucleotide. Let us just try to clear this, guys. You already know we have the purine. Now, purines are nothing but adenine and guanine, right? And we have pyrimidine. We have pyrimidine, cytosine, uracil, and thymine. Now, guys, these are nothing but the nitrogenous bases. Nitrogenous bases. Now, nitrogenous base plus pentose sugar plus phosphate. It is called as nucleotide in short. But nitrogenous base plus pentose sugar, it is called as nucleoside. Nucleoside. right? So in turn, I can simply say that nucleoside plus phosphate, it is called as nucleotide. It is called as nucleotide. It is called as nucleotide, right? Now, examples of nucleoside, guys. So nitrogenous base plus pentose sugar, it is called as nucleoside. Let us just take some examples over here. If we take nitrogenous base plus pentose sugar, okay, it is called as nucleoside. Nucleoside, right? So nitrogenous base, if we take as adenine, okay, and if we are taking the ribose as the sugar, it can be ribose or deoxyribose. So it is called as adenosine. So the nucleoside will be adenosine. These are the examples of nucleoside. Okay, adenosine. Now let us take another example. We have adenine plus deoxyribose. Deoxyribose. So it is called as deoxyadenosine. Adenosine. Deoxyadenosine. Now let us take another example. Cytosine plus ribose. It is called as cytidine. Cytidine is nothing but the nucleoside only. Nucleoside. It is composed of the nitrogenous base and the sugar. We also have thymidine. Thymidine. Thymidine, it is composed of thymine plus deoxyribose, right? So thymidine is again nucleoside. Nucleoside. So we have given 4 examples, right? There can be many as well. So this is nucleoside. Nucleoside is nothing but the sugar and the base. What is nucleotide? Sugar, base plus phosphate. Now, if we talk about nucleotide, it is more complex, okay, because it is having 3 components. 3 components, guys. We have nitrogenous base, pentose sugar, and phosphate, right? Phosphate can be 1, 2, 3, or many can be there. So let us take ATP. Now this ATP is basically adenosine triphosphate. You guys already know adenosine, it is made up of adenine plus ribose sugar, and plus we have 3-phosphate. Adenine, it is nothing but, guys, it is the nitrogenous base. Ribose sugar, it is a pentose sugar. And phosphate is phosphate. So nitrogenous base plus sugar plus phosphate, it is giving nucleotide. Okay, again we have GTP. GTP, guanosine triphosphate. Guanosine triphosphate. Now guanosine is formed from guanine plus ribose plus 3-phosphate. Guanine, it is a nitrogenous base. Ribose, it is a pentose sugar, and we have like 3-phosphate over here, right? So that is something you guys have to remember over here. Now name the types of RNA and state one function of each. Guys, you already know if we talk about the mRNA. mRNA basically carries genetic information from DNA. What is formed? RNA. And which RNA? mRNA. So mRNA is carrying the genetic information from the DNA. It carries genetic information from DNA to ribosomes. Obviously, further it will form proteins, right? Okay. So facilitating the assembly of the amino acids into proteins. Then we have tRNA. So you already know T is for transport. It transports specific amino acids, okay, to ribosomes. For what? Protein synthesis. Okay. Then we have, yes, the ribosomal RNA, the rRNA. Okay. So it is basically no structural and functional component of ribosomes. Structural and functional component of ribosomes. right? So they are combining amino acids to form the proteins, right? Other than that, we have discussed the small nuclear RNA, okay, the hnRNA. We have already discussed these functions in the previous question, right? Okay, complementary base pairing, guys, you already know about this, that it refers to the specific pairing of nitrogenous bases in DNA. Nitrogenous bases in DNA and RNA based on chemical properties. Adenine pairs with thymine with two hydrogen bonds, and guanine pairs with cytosine with three hydrogen bonds. Now, what is the importance of this, guys? Firstly, it is the stability of the double helix structure. Stability of the double helix structure, guys. The hydrogen bonds which are no formed between the adenine and thymine and guanine and cytosine, it contributes to the stability and integrity of the double helix structure. Okay, it helps to maintain the helical shape. Okay, it helps to maintain the helical shape. Apart from that, they also help in DNA replication. Now, guys, remember that one strand basically act as a template for synthesizing a new complementary strand, right? So the specificity of the base pairing, guys, the specificity that adenine binding with thymine and guanine complementary to the cytosine ensures the genetic information is copied accurately, and which is very, very important for cell division and propagation or passing of the genetic information from parents to the offspring, right? Again, there could be okay, facilitation of transcription also. Now, transcription basically means conversion of DNA to RNA. So base pairing allows RNA polymerase to synthesize mRNA, okay, based on this DNA template. And finally, we have the regulation of the gene expression. Regulation of gene expression. Now, guys, complementary base pairing basically helps in regulatory mechanisms such as binding of transcription factors. We will be discussing transcription, okay? So regulatory RNAs like the miRNAs, so they influence the genetic information as well, right? So that is like you can just take away these points which I have mentioned on the screen. So yeah, these are the various social media handles for, yeah, you can just go on for the upcoming the schedule of the live classes. Thank you for your patience and listening out.